Dear learners, welcome to the next session of solubility of drug. In today's session, our focus will be on diffusion. Diffusion is one of the important way to study release of drug from dosage form. We have seen that when drug gets released into a fluid and so as to have maximum impact, the requirement is bioavailability. So, the absorption of drug molecule from site of administration into the systemic circulation is the important characteristic to have the maximum physiological response. And during this process, diffusion plays important role. So our focus will be on diffusion, and to study this diffusion in this session we will focus on diffusion principles in biological systems, mechanism of drug transport, difference between active and passive transport, and the Ficks laws of diffusion. We will begin with diffusion principles in biological systems. The movement of solute particle or mass transfer of individual molecule by random molecular motion is called as diffusion. But the process of transfer of solute or the process of movement of solute particle is having impact of various parameters. The speed, the rate, the velocity by which this particle undergoes diffusion across semipermeable membrane, that is, the rate of diffusion is dependent on temperature, size of the particle, and size of the concentration gradient. Remember, the complete diffusion process is time dependent process, and it results in uniform distribution of solute particles in a system. The most common cause of diffusion is concentration gradient. Means the concentration has a larger impact on the process of diffusion. This common cause tends to adjust the component concentration until it reaches equilibrium. In short, we can say that diffusion is the physical flow of material. The diagram on your display shows that the transport of material from low concentration to high concentration means the diffusion process has the impact of concentration. Now see, here you are having a material residing on the high concentration node. So the movement takes place from low concentration to high concentration, and the complete process occurs until it reaches to the equilibrium. Once it reaches to equilibrium, that is the concentration outside the permeable membrane and inside the permeable membrane, when becomes equivalent, the process of diffusion will stop. Now see various mechanisms by which drug transport takes place. The principal mechanisms for transport of drug molecule across the membrane in order of their importance are passive diffusion, facilitated diffusion, ionic or electrochemical diffusion, ion pair transport, active transport, pore transport, and endocytosis. Let's see one by one. The first one is passive diffusion and facilitated diffusion. In the case of passive diffusion, the transport of molecule from higher concentration to lower concentration means the diffusion takes place from higher concentration to lower concentration. So, in this case, the diffusion is passive, or you can say that non-ionic diffusion. So, the matter moving from high concentration to low concentration along the energy gradients and does not require supplement of any additional energy. So, the driving force for the complete passive diffusion process is concentration. Whereas, in case of facilitated diffusion, this process is the carrier mediated transport system. Means facilitated diffusion operates down the concentration gradient, but this process is much faster than the passive diffusion. As like passive diffusion, again here concentration gradient is the driving force and no energy expenditure is involved. The next is active transport and pore transport. Some molecules do require energy for their transport across the membrane. So you need to supply additional amount of energy for that transport. And this is the case where the diffusion or the matter is transported from a region of lower concentration to higher concentration. That is against concentration gradient. And so as to have this travel, so as to have this movement, the energy is required in the work done by the carrier, and such processes are known as active transport. Whereas in the case of pore transport system, the process is important in the absorption of low molecular weight, low molecular size, and generally water soluble drugs through narrow aqueous filled channels, or you can say that pores in the membrane structure. So as shown in the figure, you are having a membrane which has a smaller narrow aqueous channels, or you can say pores. So the drug will penetrate across the semi-permeable membrane from these pores. The osmotic differences across the membrane or the hydrostatic pressure is the driving force for pore transport mechanism. So now we have seen that active and passive transport system. So basically one need to understand the actual difference between active transport and passive transport. So the first basic difference is the active transport is energy dependent process, whereas passive transport never requires cellular energy. The second difference that makes the difference between active transport and passive transport is the transport from dependent on concentration. Active transport occurs from region of lower concentration to higher concentration, whereas passive transport occurs from a region of higher concentration to lower concentration. For the molecules such as proteins, large cells, complex sugars, and ions, you will require active transport system. Whereas for the soluble molecules like oxygen, water, carbon dioxide, lipids, and monosaccharides, etc., you can continue diffusion by the mechanism of passive transport. Active transport is the dynamic process, whereas physical transport is the passive transport is the physical process. Comparative to passive transport, active transport is highly selective, whereas passive transport is partly non-selective process, and thus it makes active transport a rapid process, whereas passive transport is quite slow in comparison to the active transport. In case of active transport, the diffusion may take place by one side only, whereas in passive transport system you can have diffusion from both the sides. That is, passive transport is bidirectional. Now, the next mechanism by which the diffusion takes place is ionic or electrochemical diffusion and ion pair transport. In case of ionic or electrochemical diffusion, the charge on the membrane influences the permission of drug across the membrane. Molecular forms are more permeable and faster than the ionic forms. Means having the ionic form of that particular molecule will limit the permeation of the ionic forms. Out of the ions you are having, whereas you are having anion, cation, or in the some case unionized molecules, then of the ionic form, the anionic solute permeates faster than the cationic form. Means the order of permeation is cations is having least permeation, anions are having more permeation than cations, But unionized molecule, if your molecule is residing in unionized form, then it is having much faster and much permeable ratio of transportation. But likewise, in case of ion transport system, this transport mechanism stands a beneficial diffusion principle for the drugs which ionize under all the pH conditions. Despite of their low O by W partition coefficient values, the agents penetrate the membrane by forming reversible complexes. Here, if you are observing the diagram, the permeation from outside to the inside, in the first case, when you are having both the positive and negative charges, then the permeation or the diffusion takes place along with the concentration gradient, means there is no membrane potential. But the case where membrane potential observes, at that point electrochemical gradient with a membrane potential have impact on the transport. The next mechanism by which the drug diffusion may take place is endocytosis. Here, this transport mechanism involves engulfing extracellular material, means the extracellular material which is expected to be transported need to be engulfed, need to be overcome, need to be circulate by means of cell membrane. So the extracellular material within a segment of the cell membrane is pinched off intracellularly. It will result into generation of the vesicle. So in the figure, the green dots are your extracellular materials. These extracellular materials are now covered with the segment, which is resulting into formation of vesicle and thus facilitating transport, that is your diffusion. So all the diffusion process, the outcomes of the diffusion process is studied by the scientist Adolf Fick in the 19th century. Fick says that the molar flux due to diffusion is proportional to the concentration gradient, as the concentration gradient has the impact on the process of diffusion. He further states that the rate of change of concentration at a point in a space is proportional to the second derivative of concentration with space, means the process is not dependent on only one derivative. You need to focus on both the derivatives. You need to focus on the change in concentration. You do need to focus on the membrane potential, and you do need to consider the time duration for the process. There are two laws for which are the laws of diffusion given by Fick's, and that are known as Fick's first law of diffusion and Fick's second law of diffusion. Now we will move toward both the laws one by one. First of all, Fick's first law of diffusion. The molecule travel from one compartment to other compartment over a period of time. So for the time being, the rate of mass transfer can be expressed as flux, where in this equation J stands for the flux, S stands for the barrier surface, the surface which need to be overcome as a permeable membrane, which is in centimeter square. Dm upon dt is the change in mass with change in time. Fick's law of diffusion states that the flux is directly proportional to the concentration gradient, means J is directly proportional to the concentration gradient. So the mathematical representation of first law of diffusion is J is equal to minus d dc by dx, where J is the flux, d is the diffusion coefficient, and dc upon dx is the change in concentration with distance. Here the first one is your equation which represents your flux. The second equation, which is the mathematical expression for first law of diffusion process. And from these two equations we will get the third equation, that is dm upon dt is equal to minus ds dc upon dx, where the negative sign in this equation indicates the decrease in concentration with time in donor compartment. So this is the Fick's first law of diffusion. Now moving toward the second law of diffusion. The Fick's second law of diffusion states that the change in concentration with time in a particular region, either intracellular or extracellular region, is proportional to the change in concentration gradient at that point of time. So mathematically, the statement is given as dc upon dt is equal to minus dj upon dx. Now, the first law of diffusion says that the flux is dependent on concentration gradient. So, differentiating the equation 4 with respect to x, we get minus dj by dx is equal to d square c upon d square x. This will be the equation 6. As we know that the change in concentration with respect to time is proportional to the flux. That is your first law. Hence, equation 6 can be written as, considering the fact of first law, equation 6 can be written in this manner. Above equation represents diffusion in x direction only, means the movement is unidirectional. So converting this equation to the three coordinates x, y and z means making that unidirectional process into bidirectional process, we can have the mathematical expression for Fick's second law of diffusion. Thank you.